Hi, my name is Blake Porter, and I started off with case connection 1. In this case, the patient has had an enormous spike in her blood ammonia level shortly after birth, and that level only continued to rise, resulting in a drastically high amount compared with normal blood ammonia levels. What should be happening is that the ammonia should be getting cleared out of the body, or out of the blood through the liver, and then converted into urea to be peed out. But something is going wrong. Two major processes for removing excess ammonia from the body use two different ammonia acids to circulate with the ammonia and then deliver it into the liver. These amino acids are glutamine and alanine. For the majority of tissues, glutaminase synthase binds free ammonia to glutamate to form glutamine, with the previously free ammonia acid or ammonia locked into the molecule. Glutamine then moves through the body until it reaches the liver, where it is converted back into glutamate by glutaminase, removing the added-on ammonia. The ammonia, upon being freed in the liver, is now incorporated into the urea cycle to remove the ammonia from the body. In the muscles, alanine is used to control the removal of excess ammonia. Amino nitrogen is added to alpha-ketoglutarate by the enzyme L-glutamate dehydrogenase, forming L-glutamate in the muscle. Through the process of transamination, the amino is removed from the glutamate and placed on a pyruvate, which forms alanine. The alanine is removed from the muscle and moved to the liver. Here, hepatic L-glutamine dehydrogenase repeats the process of transamination, this time taking the ammonia from the alanine, which returns to being pyruvate, and adding that ammonia to another alpha-ketoglutarate, forming glutamate again, but this time in the liver. In the liver, the glutamate releases the ammonia into the urea cycle again and returns to being alpha-ketoglutarate. In the fasted state, glucose levels are reduced and the liver needs to begin gluconeogenesis to increase those levels. The recently freed pyruvate is now incorporated into this process. Pyruvate becomes glucose, which can then be carried by the blood back to the muscle tissue, where it can be used and again become pyruvate, therefore restarting the cycle over again. We know that the ammonia is traveling through the bloodstream at a very high level, so the issue is not on production or movement of the ammonia, but that the ammonia is not being properly secreted as urea. The lab tests show that the orotic acid level in the urine is also highly elevated compared to normal. This leads us to suspect that the problem is in the synthesis of the urea. Orotic acid is produced when the amount of carbamoyl phosphate is being used for the production of pyrimidine nucleotides, which means that ammonia is not being excreted as urea, but being converted back into nucleotides in the body, not reducing the ammonia levels in the blood. Tells us that the carbamoyl phosphate synthase 1 enzyme is working fine, because obviously it's producing a lot but not being getting the cycle going. The enzyme that is deficient is the ornithine carbamoyl transferase, which is supposed to react with ornithine with the carbamoyl phosphate to form citrulline. Citrulline from this point is going to become a essential amino acid and is going to have to be supplemented through the patient's diet. A genetic test of XP21.1 gene would be needed to confirm the diagnosis. The second case connection was number 3, and so gout is described in our book as the crystallization of sodium urate when the serum levels exceed the solubility limit. So this tells us what is happening, but not the cause or how to fix what is happening to the patient. Principally, the root of what is causing the patient's levels to exceed the limit and how to lower those levels to get the sodium urate back into its dissolved form. The information given to us from the patient's lab work confirms that the patient's urate level is elevated out of normal range at 10.5 mg per 100 milliliters, which confirms what we know about how the disease is being caused. Also, we get that the patient's urinary uric acid is greatly lower than normal by about half. So along with the gout, we have hypouricemia. At this point, we conclude that at least one of the options for causing the gout is wrong, the overproduction of purines. An overproduction of purines could result in the conditions for gout as a result of their catabolism process, which results in high levels of uric acid, which also results in high levels of plasma urate if they are not cleared. But if this were the case, a high number of purines would result in a high urinary uric acid count as well, which instead we have a reduced amount. The amount of sugar that the patient has recently increased in the diet could result in gout caused by overproduction of purines. The increased glucose in the cells resulting in an increased amount of ribose 5-phosphate through the mechanisms in the pentose phosphate pathway. But as previously stated, we do not see that increase in the urinary uric acid. But the labs we do get show the labs that we do not show the signs that we would expect from that being the root cause of the problem. A decreased salvage of purines could also result in increase of urinary uric acid. The process of salvaging purines involves taking the available purines and reconverting them back into their mononucleotide versions by the addition of PRPP, or phosphoribosyl pyrophosphate. An error here would cause a large supply of purines, but without the conversion back into their mononucleotide form, they would have to be catabolized into uric acid in higher than normal quantities to maintain proper homeostasis, which is again not what we see with a high plasma urate level and a lower than normal uric urinary acid. This leaves us with the possibility that the gout is being caused by a decreased urinary output of uric acid. The uric acid is not being excreted into the urine during the filtration process in the kidneys. Urate levels continue to rise in the plasma, but they are not being excreted by the body, so they just keep rising. The cause has been identified, but not the reason behind the cause. But the answer is in the patient's history. The amount of soda that the patient consumes did not play into the patient's gout by way of the overproduction of purines, but is a significant factor in the case of reducing the urinary output of uric acid. One study by Dr. Hyun Choi of the University of British Columbia showed that sodas could raise the risk of gout by 35% per serving. Explanation could be deduced from another paper, whose explanation is that the SLC-A9, a known transporter gene, is also responsible for the transport of uric acid. So we have two compounds that both need to be excreted in their excess, the uric acid and fructose. The quantities of soda and its fructose ingredient have probably overwhelmed the transport to the point that it can't catch up with transporting both uric acid and fructose.